I'm going to give a couple. My name is Jason Heindel, and you've probably gotten some emails from me. And I'm going to give a couple slides about the branch. Things are going crazy. There we go. And hopefully encourage you to become a member of either the branch or of the national chapter of ASM if you're not already a member. One thing you get to do when you're a member, or you don't have to be a member, but you can go to ASM Microbe, the annual meeting. This year it's in Atlanta, June 13th to June 17th. Some of us in this room will be there, so you can see us there or avoid us, whatever you choose. And we also want to say congratulations to the Peg Cotter Award winners for this year. So all three of these individuals will be at the meeting. So Lauren Cadiz, I don't know if I'm saying that right, Aaron Boulanger, and Kyle Rodino. So congratulations to them. We're very excited to bring these young scientists to sponsor them and have them present at the meeting. So it's a really great opportunity for them. And next year, when you see the info come out about nominations or applying for this, if you are eligible, you should do so. That's a great opportunity to get a good amount of money to go to this meeting, which I think will be in Los Angeles in 2 years. All right, so for the branch, what do? We have a number of things that we focus on, including these monthly meetings where we have speakers brought in from all over the country or from right here in Philadelphia. We also have an annual I&I forum, which is held in December usually. And we have a bunch of outreach and education programs, and we have an annual clinical microbiology symposium that is also very popular and well attended and an important resource for the local microbiology community. So you should consider engaging us in all of those aspects. 2 upcoming meetings. So in next month in March, we're hosting Cami Lesser from Mass General Harvard Medical School. She'll be giving a talk, probably in this room or a room close by. And then in April, we're hosting Joanna Engel from UCSF. She is a distinguished lecturer of ASM. So we're getting some assistance from national chapter to bring her all the way out here from the West Coast. This will both be great meetings. And then in May, the student chapter is organizing a monthly meeting, and we have a Norm Willett Memorial Lecture also upcoming. So stay tuned for more details on both of those. We also have a new website that is about to go live. So if you get an email saying, hey, go get your login credentials, do so. There's going to be some new functionalities and member-only content. So this will be very exciting and hopefully very functionally relevant for us. Finally, talking about membership, it's not expensive, particularly if you're a student or an early career scientist. In fact, for the branch, to join the branch here, if you're a student, it's free. So you can't get better than that. National membership is only $35, which is for an annual membership. That's a good affordable price. And the prices for other members on the right-hand side. We encourage you to join both. We really encourage you to join the branch at least. But if you join both, that's great too. Membership is on a calendar year, not an academic year. And it does not roll over automatically. And you may not even get reminders, or you may delete those. So if you think you forgot to renew, check it out and go ahead and renew. All right. So now I'm going to give a quick intro to our speaker, who I think many of us have we've at least seen the papers. We've interacted with him in other venues, but I'm very excited that Christophe accepted our invitation to come down the street here and talk to us. He did give me his bio, which is very nicely written. Thank you. So the important information is he's currently an assistant professor in the Department of Microbiology at Penn. But prior to that, he did his undergraduate studies at Bonn in Germany. I did a master's of science studies at Yale and ETH Zurich. And then he did his PhD in Rehovot, Israel at the Weizmann Institute, and had a visiting fellowship at Stanford as well. So he's had a number of really prolific and high-profile background already. He also spent a little bit of time, which maybe I'll hear about over dinner, at the Broad in Cambridge, doing some work there before he came in 2018 to Penn as an assistant professor. So relatively new. The most important information, I think, is that pretty much every time a new award comes out, he's either a finalist or a recipient of that award. And every time a really new, interesting, exciting paper related to the microbiome and its effects on human physiology comes out, he's typically one of the authors on that paper, or 20 of the references are from his lab's work. So it should be quite exciting to see what he's going to talk to us about with microbiome aging. So thank you. Thanks.
Great. Hi, everybody. Thanks, Jason, for the nice introduction, for inviting me to be here today. I just calculated while Jason was talking how many years this must have been going on if I'm the speaker number 808. So that's really, really impressive. And what I'll talk about today is I'll start with an overview of everything we do in the lab, and then I'll show you a few highlights of studies that have been recently completed or that are ongoing. But everything we do in the lab is focused on the goal of trying to identify commonalities of the major human diseases you see here on the slide, which are, very broadly speaking, neurodegeneration, cancer, chronic metabolic disease, and inflammatory disease. And of course, the reason why we're so interested in identifying commonalities is because we think there are ways to act much, much earlier in the prevention and treatment of these diseases, if we understand the joint mechanisms in the pathology of these diseases. What's really hard, though, in trying to understand these commonalities is that it's not clear what the physiological counterpart is. Usually when we think about an infectious disease, it's very easy to rationalize from the standpoint of evolution because there are two players, they try to outcompete each other. But here it's unclear what the evolutionary process is, what the physiological mechanism is that has been selected for. But one possible answer is what's called the environmental mismatch theory, which basically says there has been selection, but the selection occurred during a history of human evolution, which was completely unlike our current environment. And now we're exposed to foods and other environmental elements that are completely different compared to human evolution. And that's why we're now more susceptible to these diseases. So that's something we study very extensively in the lab, by viewing a human being as the product of all of these environmental factors that we're exposed to. So each bubble here, when I started the lab a few years ago, each bubble here was one of the topics that a new student or a new postdoc picked when they joined the lab, trying to identify new mechanisms by which all of them, either in isolation or jointly, influence our individual susceptibility to disease. And just to get started, I'll highlight a few of these environmental factors that we're very interested in, starting with what I think is by far the most beneficial. And this will not be a surprise to anybody, but it's the environmental factor down here, which is exercise. Because if you think about the diseases we just looked at the first slide, there is now unanimous evidence in the literature that exercise is basically the silver bullet we have that protects us across the entire board, ranging from neurodegeneration to cancer, metabolic disease, and everything. And this is based on many, many studies like this one here, where people have looked at essentially the risk for mortality against the amount of medium level intensity exercise that people carry out every week. And you can see here, you can look at all-cause mortality, you can look at cardiovascular-related mortality or cancer-related mortality. The curve essentially looks the same for each of them. There's a very steep decline early on, and then it plateaus. And this steep decline is better for exercise than for any other environmental factor that we know of. It's better than any drug, better than any diet, everything that has been studied. This is a huge, huge reduction here in all-cause mortality. And then at 150 minutes, it plateaus. So 150 minutes of moderate level intensity exercise is what we should all be doing every week in order to leverage these benefits. And of course, the problem is that globally speaking, we're extremely far away from reaching this WHO recommendation. And this is not only a global problem, but just this morning I checked the statistics for Philadelphia, and Philadelphia is roughly on par with the global average and really far away on the population level from reaching these 150 minutes of exercise that we should be doing every week. And so we got really interested in this phenomenon, and we started studying this a few years ago, specifically a very really fantastic PhD student in the lab, Lenka Donalova. And what she found is this pathway described down here, which told us that the motivation and the drive to exercise is not necessarily only brain intrinsic, but actually has a large component outside of the brain, specifically originating in the gastrointestinal tract. So what Lenka found is that there is a pathway that originates with the microbiome and specific molecules, fatty acid amide molecules that the microbiome is producing. And these molecules send a signal to the brain through this pathway that includes sensory neurons, TRPV1 positive sensory neurons, and then influences dopamine homeostasis in the striatum by down-regulating the negative regulator of dopamine, which is monoamine oxidase. And more dopamine during exercise drives the motivation for more exercise. This is basically the pleasure and euphoria we get from strenuous exercise, which we sometimes call the runner's high, this is largely mediated by dopamine in the striatum, the motivation for movement. And we basically arrived at this conclusion through a few experiments that I'll show you now. The first one is this one here. This was an experiment we did together with Nick Beckley, who's running a neurophysiology lab over at Penn. And they looked into the striatum of an exercising mouse using fibrophotometry of a dopamine sensor. And you can see here when the mouse is exercising, you get this huge surge in dopamine, just like in humans. What was really interesting, though, was when we repeated the experiment and we ablated the microbiome, essentially the most upstream component of this pathway here, now there's a flat line. So that dopamine surge is not brain intrinsic, but requires an intact microbiome. And then we did a few more experiments, which I'll show you next, which is asking for causality of this. So we did experiments where we basically block downstream, stimulate upstream, and expected the mice to run. Or we stimulated downstream, blocked upstream, and expected the mice not to run. And this is what I'll show you now. So first, I'll show you that if we ablate the most upstream component, again, antibiotics, and then we put a mouse on a running wheel, its motivation to run is drastically reduced. But when we stimulate these sensory neurons here, unspecifically using capsaicin, which you will know is the ligand for TRPV1, we can now restore exercise even in the absence of the microbiome. So now the mice run just fine, even though we ablate their microbiome with antibiotics. But if we then come with another drug, sulpiride, which blocks dopamine receptors in the striatum, now running is again down to the antibiotics level. So we can basically cancel the stimulation by blocking upstream. And we can do the opposite experiment, which uses a mouse called the TRPV1 DTA mouse, which is lacking these sensory neurons. This mouse doesn't like to exercise, just like antibiotics-treated mice, but we can make them exercise just fine as long as we stimulate their dopamine receptors using yet another drug, which is called bromocriptine. So this told us that the dopamine aspect must be downstream to the intestinal manipulations we do with the microbiome. And then I'll just show you two more pieces of data from this study, which is that we can leverage this by, again, bypassing the microbiome and just supplementing these fatty acid amides, which we found to be produced by the microbiome. So we designed a diet that contains these molecules. We call this the FAA diet. And now you can see that even when we treat the mice with antibiotics, the mice run just fine. So there's nothing intrinsically wrong with the absence of the microbiome as long as we provide these fatty acid amide molecules. And then dopamine is restored, running is restored, and the whole pathway depends on this molecule here, the endocannabinoid receptor CB1, because if we use a pharmacological blocker of this, now the beneficial effects of these FAAs are gone. So this led us to conclude that this FAA production by the microbiome binding to CB1 and then triggering this upstream pathway is what's important for exercise motivation. And the very last thing I'll show you on this study is our efforts to try and identify what this arrow is here. How does the microbiome produce these molecules? And here we actually had a lot of help from a group at MIT that had just figured out the biosynthetic pathway by which the microbiome produces these fatty acid amide molecules. And they identified a biosynthetic gene cluster that is present in specific members of the gut microbiome, by which those members of the microbiome can produce these molecules. So we took one of these members of the commensal microbiome, for example, Eubacterium rectale, and we cloned out these genes, this biosynthetic gene cluster, which we knew from that group was responsible for fatty acid amide production. And we cloned it into a bacterium that doesn't have it naturally, for example, E. coli. So we generated this engineered strain of E. coli that now is now endowed with this gene cluster. And what's really interesting is that this engineered strain now starts producing fatty acid amides, for example, OEA. You can see here in a germ-free mouse, there's almost nothing. If we colonize the germ-free mouse with the parent strain, there's still nothing. But if we colonize it with this engineered strain, now it starts producing OEA. And this by itself is enough to make the germ-free mouse run more, which is what you can see here. Germ-free mouse, as expected from the antibiotics experiment, exercises poorly. But if we can boost this, if we put in this OEA producing strain. And again, this beneficial effect is dependent on the CB1 receptor, because if we now co-treat these mice with AM6545, which is this drug that blocks CB1, you remember from the previous slide, now this beneficial effect is gone. So this led us to the conclusion that there is a very important contribution of the microbiome in driving the motivation for exercise. And that's, of course, important because exercise is such an important factor in controlling overall health. Now I'll switch, for the next few minutes, I'll switch from what I think is the most beneficial environmental factor to what's probably the most detrimental environmental factor in terms of overall health. You can take a guess which bubble this might be, but epidemiologically, we know that psychological stress, which is up here, is associated with the most strongly exacerbated disease outcome and the poorest performance in terms of long-term survival. And we know this from studies like the ones that I'm going to show you in a minute, but first I'll show you the people in the lab who are working on this topic. Markus Schneider actually just started his own group back in Germany. Niklas Blank is a PhD student in the lab, and Jelena Alvarez was a gastroenterology fellow at Penn. She's now at the Rockefeller, and she told me that whenever she does colonoscopies and she has a chance to talk to the patient before or after the procedure, she sees a very strong correlation between their levels of stress and the outcome that she sees by colonoscopy. And of course, it's not only Jelena's private observations, but there are studies like this one here, which link the outcome of IBD patients in this case with self-evaluated stress levels using a questionnaire. And there are many, many, many examples like this one, not only in IBD, but in many other inflammatory diseases where we know that high levels of psychological stress are associated with poor outcome in the long term. But the mechanisms are largely unclear. We don't know, in most cases, we don't know how a brain-derived signal, like the perception of stress, is communicated to the periphery to drive an inflammatory response. So we worked for many years on a pathway I'll describe in a summary cartoon here, which is linking stress perception by the brain to the canonical HPA axis, which, as you know, results in the production of glucocorticoids in the periphery. And then it gets interesting because these glucocorticoids, they now act on neurons and glia, not only in the brain, but neurons and glia in the enteric nervous system. And they have a pretty strong effect on shaping the inflammatory profile of these glia cells to produce CSF1 and many other inflammatory mediators, which then recruit monocytes. And monocytes start producing TNF, which is a known driver of disease exacerbation in IBD. And at the same time, we found interesting effects on enteric neurons, which lead to the basically dysmotility and obstipation in these individuals. So I'll show you again, just like with the exercise project, I'll just show you a few examples of experiments we did that led to these conclusions. The first one came from single cell profiling of enteric glia cells during episodes of psychological stress in a mouse. So we stressed mice in various different ways. We took their enteric glia cells from the gut and we identified 4 different subsets. And one of those subsets, you can see here on the right side, was only present under conditions of psychological stress or dexamethasone, which is stimulating glucocorticoid receptor signaling. And so Markus called this subset enteric glia associated with psychological stress, or EGAPS. And now with the identification of these cells, now we can ask what's the function of these cells? What are they doing in the gut of a stressed mouse? So one of the tools we used was basically one of these interaction networks you can derive based on single cell RNA sequencing data sets. And this interaction network told us that these EGAPs are talking to monocytes and other myeloid cells in many different ways. And this was interesting because we had observed that during episodes of psychological stress, there's an accumulation of monocytes in the GI tract of these mice. You can see this here on the right side. Those of you who are immunologists, they will know that in GI myeloid cell biology, there is a phenomenon called the monocyte waterfall, because there are monocytes that are recruited from the systemic circulation. They are up here, Ly6C positive, and then they differentiate with a lot of imagination. You can see a waterfall going down here, basically to the macrophages that are MHC2 positive but Ly6C negative, and these are generally anti-inflammatory. But now in the stress condition, the waterfall is broken, and you can see here that many cells accumulate up here where the monocytes are. So that was interesting, and it was even more interesting in the context of this finding here that there are factors like CSF1 produced by these inflammatory enteric glia cells, because CSF1 is a known recruitment factor for monocytes into the gut. So we did an experiment where we blocked CSF1, and here's the outcome. You can see that this is again what I'm showing you up here, that in a stressed mouse we have more of these monocytes, But this is entirely dependent on CSF1, because if we block CSF1 with an antibody, we can stress the mouse as much as we want. There is no influx of monocytes. And what's even more interesting is that this completely mirrors the disease phenotype we see. So if we, for example, we profile the mice by colonoscopy or many other ways to look at colitis outcomes, you can see that colitis is much worse in the stressed animals, but not if we treat them with an anti-CSF1 antibody. So we can rescue the effect of stress on colitis through this treatment. And we can also do it by just ablating monocytes, which is what we did here with CCR2-DTR, with a genetic ablation of monocytes, or by ablating these inflammatory enteric glia cells, which we do here using a SOX10 driver and again the diphtheria toxin system. So here in all of these cases, even though we stress the mouse, it doesn't respond with a stronger disease severity in terms of colitis, which meant to us that this pathway here that links stress perception by the brain, this inflammatory profile of enteric glia and monocyte recruitment is as a main driver of this. And now we're looking at this in a clinical trial as well, because we have some promising early data that the same pathway is active in humans, where we can basically score someone's levels of stress using this questionnaire I've shown you in one of the previous slides. And then we can see if there is an opportunity to block some of these molecules like CSF1 to see if we can protect these individuals from the negative consequences of psychological stress. But for the rest of the talk, I want to highlight another big topic, because this environmental mismatch theory is only one theory to explain these many different diseases and why they're rising so strongly in the human population. As I told you before, the environmental mismatch theory is based on the idea that selection has happened evolutionarily, but during a different period of human evolution. But there's a second alternative explanation, which is that all of these diseases are diseases of older age, because as we age, our susceptibility for these diseases gets higher, and that selection is happening even in present-day human life, but it's happening after the reproductive period, so it's not effective in terms of protecting us from the detrimental effects of these diseases. And so we got very interested in this because aging is by far the highest risk factor for anything from neurodegeneration to cancer to chronic metabolic and chronic inflammatory diseases. And also because there seems to be, just like exercise, there seems to be another silver bullet we have to protect us against the detrimental effects of aging, which is fasting or caloric restriction. I'm sure many of you have heard about all these many fashionable trends of eating less at different times of the day, or eating less overall. And you can see here that the most popular one is what's called the 16-8 method, which is basically you eat only for 8 hours of the day, and for 16 hours you don't eat. And there are many variations of this theme. Some of them are you basically pick 2 days of the week and you eat less, or you don't eat at all, which is this method, what's called the eat, stop, eat method. You can do every other day fasting, where you don't eat every other day, or you eat fewer calories every other day. Then there's what's called the warrior diet, where you basically have one big meal sometime in the afternoon, and during the rest of the day you can eat very little and only fruit. So there are many different variations on this overall approach. And in animal models, this is extremely effective in terms of extending lifespan and promoting health. For example, here is a study from 2019 where they basically put mice on dietary restriction. And you can see the red curve lives much longer than the blue curve, which is ad libitum fed mice. And you see the benefits even if you switch the mice to ad libitum late in life, or you start fasting them late in life, there's still some benefit. And of course, there's hundreds of papers by now in flies and C. elegans in many different model systems. It extends lifespan and promotes health. There is even a study in monkeys. I don't have the labels here, but you can clearly tell which monkey looks. I'm not sure if this monkey looks happier, but it certainly looks healthier overall. And actually, in humans, the people in the aging field always talk about the fact that there are no studies in humans yet, so we don't know if fasting extends life in humans, but we know it will certainly feel much longer. And I think this is what this monkey is probably going through. But so this is a very compelling example of how caloric restriction has long-term benefits, even in species that are closely related to us. The problem, though, is that there is something that is common to all of these studies, which is not always publicized very strongly. but there is a giant variability from individual to individual in terms of the overall health effects. And this is most dramatically illustrated by this study here, which has looked, again, in mice, more than 10 years ago by now, but mice of both males and females, and mice of very heterogeneous genetic backgrounds. And you can see that there are some mice that have a very strong health benefit from and lifespan benefit from dietary restrictions, but others don't. Others actually die early. And this is, of course, really concerning for thinking about human applications of this, because humans are genetically heterogeneous. And there's no good study on and it's also very hard to study because the study will be extremely long, but there's no good knowledge about how effective this will be in humans. So we a few years ago we started a very big study trying to understand what regulates this heterogeneity in terms of individual responses to caloric restriction. And this was a study that was so large that we weren't able to do it all ourselves, but we teamed up with the Jackson Lab, and specifically Gary Churchill. The Jackson Lab, as you know, is this very big mouse vendor up in Bar Harbor, and they can do these very large studies of thousands of animals over the lifespan with many different dietary interventions. Another partner was Calico, which is Google's life science company that is also very interested in aging. And on our end, this was led by Lev Lidichevsky, who's an MD PhD student in the lab, just about to graduate, but he's been absolutely fantastic. He's doing mostly computational work, and he's been co-advised by Mingyao Li, who is a biostatistician at Penn. And I'll show you what the experiment looks like. The experiment is almost 1,000 mice, genetically diverse mice. So each mouse is genetically unique. We generate these mice by basically crossing mice with a known genetic background. So there are 8 founder strains, and then we generate essentially infinite genetic diversity over multiple generations. And then they go on 5 different dietary protocols, if you will. So there is ad libitum, the mice eat all the time. And then there is this very similar to the slide I've just shown you, the mouse is not eating for a day, or not eating for 2 days, or eating every day, but 20% less or 40% less. And the bottom line, I'll say it right away, is that it works extremely well. So the mice are living longer, as we would expect from all these previous studies. So even though these are genetically heterogeneous mice, if we basically fast them for 1 or 2 days, they live longer. If we fast them 20% every day, they live longer. If we fast them 40% every day, they live even longer. And so that's a sort of encouraging outcome. Just on the side, as you would expect, these diets have very profound effects on physiology. Here you can see the weight trajectories of these animals over their lifespans. Normally a mouse sort of grows until around 20 months of age roughly, and then starts losing weight. Most of the different diets do the same, but with a reduced overall weight. But the 40% caloric restriction, they basically crash, and they are very lean throughout their entire life. And you can plot this, just because these mice, the red group, lives much longer, you can plot this as proportion of life lived. So you rescale the axis, and then you get rid of some of this survivor bias. But the point I want to make here is that even though overall these diets all work in terms of extending life, it's still the outcome is extremely, extremely heterogeneous. So you can see here, this is basically the lifespan of each individual mouse. And here there are some mice on the 40% caloric restriction diet, they die as early as the earliest ad libitum mice. And of course, you wouldn't want to be this mouse because it's hungry every day, it's extremely lean, and yet it dies young. Right, so there must be something that regulates this extreme heterogeneity. They don't commit suicide, but and you know, to be fair, we don't always know the cause of death. Sometimes we just find them dead, and then sometimes we can perform an autopsy to identify the cause of death, but in many cases we don't know. But these mice are, I mean, they must be miserable, right? They're sick for some reason, and they're definitely extremely hungry. So we set out, as part of this very large study, we set out to identify the reasons for this very strong heterogeneity. So what we did is we tried to profile and phenotype everything we possibly could about these mice. Of course, what's most important to us is we profiled their microbiome longitudinally over the course of their lives. But we also got a lot of other information on, metabolic cage data, immune data, frailty, metabolic phenotyping, glucose, CBCs, and so on. And then we took all this data and tried to identify where the variability comes from. But first, I'll show you the microbiome. And of course, the microbiome is extremely interesting in the context of a large study like this, because we can, so we metagenomically sequenced all these thousands and thousands of samples, huge effort by Lev and other people in the lab. And then we can plot all these microbiomes on a classical PCOA axis, where each dot is a microbiome, and then we can relate it to all the meta information we have. And you can immediately see that both diet and age expectedly are very strong drivers of where the mouse will end up on this PCOA plot. But then it got really interesting because as part of this study, remember, each mouse is genetically unique, and we can actually genotype them because we can perform SNP genotyping, and we know the founders' genomes. So we can genetically, basically we can assign a host genome to each dot here. And this is interesting for two reasons. One is it allows us to do a pretty interesting trick that Lev worked on extensively, which is never done usually in mouse microbiome studies, but we can identify the mouse DNA based on the fecal metagenomes that we sequence, because there is some mouse DNA in the feces. And as part of our sequencing process, we get the mouse DNA, and then we can see if this matches the DNA that we expect. So we get plots like this one, where we basically calculate how similar the expected mouse genome is to the observed mouse genome. And then we get curious cases like this one here, where this is a mouse ad libitum fed mouse 97 at 44 weeks. Looks exactly like ad libitum mouse 97, so everything's fine. And here we have another mouse ad libitum fed mouse 104 at 44 weeks, 105 at 44 weeks. Looks exactly like the mouse we expect. But then something happened, because now a few weeks later, this mouse no longer looks like itself, but it now looks like ad libitum mouse 105, and ad libitum mouse 105 looks like ad libitum mouse 97. So what must have happened is we must have swapped the tubes or the mice or the collection process. And there was very strong evidence, because we have longitudinal body weights from these. And you can see here that the mouse now, all of a sudden, in our recorded body weights, it jumps down and the other mouse jumps up. So these were clearly swapped. So that's a very expensive exercise to identify where we did swaps by metagenomically sequencing 3,000 samples. But what it allows us to do is we can retrospectively correctly assign the mouse that we collected from, but also we can calculate the impact of the host genome on these 3,000 microbiomes that I've just shown you. And we can compare it to all the other meta information that we have. And the result is this plot here, which is basically ranking all of these different factors according to the proportion of variance in the microbiome explained. And very unexpectedly, genetics is number one. It's at least on par as some of the other ones, like diet and age, that we knew to be a strong driver of microbiome changes. And the reason why this was so unexpected is because in humans, where we could have, where we've been able to do these kind of studies for 10 years using basically, genome-wide association studies for microbiome species abundance, by having a microbiome sequenced and a host genome sequenced, the unanimous conclusion from these studies was that the genetic contribution to microbiome composition was extremely low. So we took everything from the literature we found about these studies, where people have calculated percent of taxa that are heritable, and we found that in humans, the estimation is generally low. In agricultural animals, it's somewhere in between, and in genetic diversity mice, the estimate is much higher, which probably means that the housing conditions matters a lot in terms of bringing out the heritability effect on the microbiome, because human lifestyles are chaotic and uncontrolled. In agricultural animals, it's more controlled, and in mice in cages, it's extremely controlled. There's very little room for other variables. So that's why we think we see the genetic contribution on microbiome heterogeneity much more strongly than in human studies. But it also means that the theoretical possibility for genetic control of the microbiome is there. It just hasn't been observed because it was primarily done in human studies. There was a second really big surprise when we looked at this that I'll show you next, which is when we look at the aging effect on the microbiome. You've already seen on the PCOA plot that there was a very strong shift with age in all of these mice, globally speaking. And this is reflected here if we look at the taxonomic and functional units of the microbiome. Everything that's in green is significantly affected either up or down by age. So you can see that the big majority of elements that we can quantify are affected by age. So age is a very strong driver of microbiome changes. And as a consequence, not surprisingly, we can predict the age of the mouse by sequencing its microbiome. That's again not the purpose because it's a very expensive way to tell the age of the mouse, but it allows us to do comparative studies of our study of this genetically diverse cohort to other mice, like Black 6 mice, that we profiled along the lifespan in our lab, as well as humans from a very large database where we knew the microbiome composition and we knew the age of the donor. And you can see that in each of these cases, there's a very strong agreement of the percent features associated with age, 60% in both taxonomic and functional levels. This means that both in mice and in humans, there's a very strong impact of age on the microbiome. But the big surprise came when Lev compared the taxonomic elements that are affected by age in all these different studies, which is what you can see here. There is basically no agreement. The trend line, the gray line, is always completely flat, which means that strains that change with age in a mouse are different from strains that change with age in a different mouse compared to strains that change with age in humans, with only one single exception. If you look here, the top, basically the right quadrants of each of these graphs, you see that there is something called uniqueness again and again and again. Uniqueness here, uniqueness, uniqueness. So what is uniqueness and why does it go up with age? Uniqueness, here you can see it go up with age. In the diversity outbreak cohort, like 6 mice over the lifespan, humans over the lifespan, uniqueness goes up. It's the only thing that reproducibly goes up with age. So what is uniqueness? Uniqueness is basically the dissimilarity of any microbiome to its neighbor in a PCOA plot, for example. So if we plot them in the PCOA space, they distribute in a certain pattern, and then uniqueness quantifies the distance from each dot to the nearest neighbor. So if uniqueness is very high, then that's basically a dot down here that is very far away from the nearest neighbors. And here in this huge crowd, the uniqueness is very low. So what we found is that on the left side, both in mice and in humans, uniqueness goes up with age, which means that as we get older, our microbiomes become more and more dissimilar from other individuals' microbiomes. So now the question is, does uniqueness go up with age as a function of the age of the host or as a function of time? Is there a determinant of an old gut that produces a high uniqueness, or is it going up because time passes? And Lev found a way to test this experimentally, which is the following experiment. He calls it the co-housing experiment. So here we have young mice in blue, old mice in red. And then he used some of these young and old mice and he co-housed them in the same cage, which, as you know, will equilibrate their microbiome because they start eating each other's feces. And then after a certain period of co-housing, he separated them again. And the idea here is that we can basically tell what's stronger, the effect of time on the microbiome or the effect of the age of the host, because we disentangle the two by co-housing them. And here you can see the result. First, here are young and old mice. Blue and in red, as you would expect, they're very different at baseline. And then we co-house them, and now the co-housed young mice and the co-housed old mice look like old mice initially. So they're basically, the aged microbiome is dominantly transferred to the recipient mice once they live in the same cage. And now the interesting question is, what happens if we separate them again? Do they look like old mice or do they look like young mice? And that's equivalent to two theories, basically the selection theory versus the neutral theory, which is often used in evolution. The selection theory would posit that it's the age of the host that is basically selecting for a specific microbiome, in which case we would expect a young mouse to reverse its microbiome appearance to a formerly young microbiome, versus the neutral theory, which says that it's all stochastic and a function of time, and the age of the host doesn't matter in terms of bringing it back. So what I'll show you next is what we think is the solution, but does anybody want to take a guess if it's purely time, or if there's maybe everybody raise their hands if you think it's the age of the host that matters. Okay, 5 people, and I guess the rest thinks that it's a function of time. Who thinks it's just random? Okay. So I'll show you the result here. So basically what happens is that this is a young mouse living by itself at baseline. And this is the distance to itself, essentially. So it's very similar to itself, which is expected. The old mouse, you can see here, has a distance that is basically the distance from this centroid to this centroid here. Now we co-house it, it becomes very similar to the old mouse, very dissimilar to itself, And now we separate it and it never goes back to normal. So the majority had a good instinct here that it's not the age of the host, but it's actually the time that passes. And this is more strikingly demonstrated here by looking again at uniqueness. So you already know that between young and old mice, uniqueness is different, right? Uniqueness is higher in the old mice at baseline. And then you co-house the young mouse and it acquires a uniqueness that looks like an old microbiome. And when you separate them, it never goes back down. So the microbiome of a young mouse is plastic in the sense that it can acquire a high uniqueness and it's irreversibly acquiring this high uniqueness. And to me, the most striking demonstration of this is when Lev trained a machine learning algorithm based on baseline samples that tells us with 100% accuracy if a mouse is young and old. This is basically what you've seen on the previous slide, this age predictor based on the microbiome. But now after co-housing, the predictor cannot tell a young mouse from an old mouse anymore. It thinks that the young mouse is old because the microbiome looks old, and the old mouse is still identified with 100% certainty. So basically, this means that most likely what's happening is that there's a stochastic process of microbiome aging. Time is extremely important. That's why aging has such a strong impact on the microbiome, but there is no deterministic effect of the age of the host, which is why microbiome aging looks so different from one mouse to the next, from one cohort of mice to the next, and from mice to humans. So now the final question is, if there is a stochastic process of microbiome aging, does it help us with our original question, which is, can it explain some of the heterogeneity in longevity and dietary restriction outcomes? So that's what we asked next. So first, I'll show you a summary of all the associations that we looked at. So these are different health parameters, metabolic parameters, immune and hematologic parameters, and microbiome parameters. And then we look at how they're affected by body weight, by diet, by percent of life lived, which remember is this scaling of lifespan, or by the interaction of both. And whenever there is a significant hit, we color it in orange. And you can see there's a lot of orange in the microbiome, which is what we would expect. Very strong associations of the microbiome with diet, weight, and many of these other things. But then we look at associations with lifespan. And now everything in the microbiome turns green. So we didn't find a single significant association of these microbiome changes with lifespan outcomes, which is yet another argument for assuming that the microbiome development over the lifespan is largely stochastic. It ends up with a high uniqueness reproducibly, and it has many associations with host phenotypes, but not with lifespan. While other things do have associations with lifespan, interestingly, primarily hematologic and immune components. And for the last 5 to 10 minutes, I want to show you if the microbiome does not associate with lifespan and dietary restriction outcomes, then what does? So we performed what's called a partial correlation network analysis, which basically shows us out of all of these different traits that we measured in the mice, how strongly correlated are they to each other and how strongly correlated are they to lifespan. And then we can color everything in red that is significantly associated with lifespan. Now, lifespan is this one here, which sits just in the middle of the graph. And around it, you can see there's basically this patch of colored circles, all of which are predictive of lifespan. And then there are others that are far away and are not predictive, are strongly correlated with each other, but not predictive of lifespan. So now the question is, what are all these traits that are associated with lifespan? And do they determine a mouse's response to dietary restriction? So to do this, we used this what's called an onion plot here, which is basically telling us how diet affects many of these different things that we measured and how these different things that we measured associate with lifespan. And then we can go one by one by one by one, and we can try and identify these different mechanisms. But I'll first show you that this is a very powerful approach. For example, if we just use this one here, which is body weight, and we stratify the mice by body weight, which is what we did here. We divided them into light mice and heavy mice at the time of randomization. You can see that the outcome by just doing this stratification is extremely dramatic, because a light mouse would support our overall conclusion that all of these dietary interventions extend the lifespan of this mouse. But if you're a heavy mouse and you go on a 1-day or 2-day fast and you end up on the light blue or the dark blue curve, it doesn't help at all. So a heavy mouse needs to go on a 20% or 40% caloric restriction in order to get any health benefits. And we can do this now for every single trait that we measure, see whether it extends lifespan and whether it determines the responsiveness to these different diets. So now let's go through some of these pathways here along this onion plot just to demonstrate what I just told you. So predictor number one, pretty surprisingly, was lymphocytes. So lymphocyte counts go down with age. This is very well known. There's many of you will know about this, the myeloid skew that happens at old age. So the lymphocyte numbers go down. The proportion of myeloid cells go up. And you can see that what the diets do is they counteract this trend. The gray one here is the ad libitum group, and the diets basically push up the number of lymphocytes. And this is very beneficial because across all of these different dietary interventions, if you have a high lymphocyte count, the mouse is more likely to live longer. So high lymphocyte count at old age is good. The second example, which is a little bit counterintuitive, is body fat. So body fat also decreases with age, both in mice and in humans. And you can see here that what all these different dietary interventions do is, of course, they reduce body fat, right? If the mouse fasts, then body fat is reduced. But this is actually detrimental because the mice with low body fat, they die younger than the mice with high body fat. So here we have an example where the diets extend overall lifespan, but with this specific example, the mice that actually withstand this effect of the diets on lowering body fat are the ones that live longer. And I'll show you one more example, which is, I think, the most striking one, which is on erythrocytes, and specifically on RDW, which is red cell distribution, which is one of the parameters we get in a blood CBC. And basically what this RDW describes is the distribution width of the size distribution. So erythrocytes, as we get older, they tend to be more diverse in terms of sizes. So we have smaller erythrocytes and very large erythrocytes, and this means that the distribution of the volume of these erythrocytes gets larger as we get old. This is what you can see here. The RDW goes up with age. Really interestingly, all these diets increase in RDW. So you get more red cell distribution when you are on a fasting diet. And this is, again, really detrimental for health because you can see that the mice live much longer if they have a low RDW. So this is yet another example where the diets are doing something which is apparently detrimental, but their overall effect is extending lifespan. So I think we have basically a composite picture here where the diets have all these different effects. Not all of them are lifespan promoting, but some of them are actually counteracting lifespan. And the mice that live the longest are the ones that basically select the beneficial pathways out of this onion plot and manage to avoid the detrimental ones. So for example, the mice that have the highest lymphocyte count, but the highest body fat, but the lowest RDW, these are the ones that are these ultra long-lived mice in the different groups. Now, one last example, because this was also pretty surprising, is that you can see here that the glucose shows up on this plot, but has very, very tiny arrows in all directions and no direct arrow to lifespan. And this was very surprising because glucose is, of course, the strongest responder to dietary intervention, the one that we measure in humans all the time because we think that that's the strongest benefit we get from fasting. And we get it in the mice. You can see here that the ad libitum mice are by far the ones with the highest glucose levels. With all these different diet interventions, we reduce glucose, but it doesn't matter for longevity at all. Which, you can see here on these plots, none of these p-values are significant. This one gets close, but once we correct for multiple testing, it's also going away. So glucose levels in a mouse, at least on different dietary interventions, doesn't matter for lifespan. So with this, I'll summarize this study that the host genome has a surprisingly large impact on microbiome composition over the lifespan. And there is no universal signature of aging, but there is this consistent increase in uniqueness. And we think this consistent increase in uniqueness is a function of, basically, it complies with the neutral theory. So it's a function of time. There's no selection for it. And we get it in many different microbiome trajectories over the lifespan, from human to human, from mouse to mouse, and so on. There's a very strong impact of the microbiome on health, but not on longevity. But longevity is regulated by these non-overlapping regulators of metabolic health and longevity. And some of them have these surprising antagonistic effects, like these, the pro-longevity factors of dietary restriction on lymphocyte counts, for example. But there are these anti-longevity factors on RDW, adiposity, and many others that I haven't shown you. But as far as we can tell now, after analyzing all this data, basically the best mouse is the one that manages to select the beneficial effects of a diet, has the right predisposition in terms of body weight and starting conditions, and manages to avoid these anti-longevity factors on RDW, adiposity, and some others. So I'll stop here, but of course I want to show you everybody who was involved in all of this work. As you can imagine, this was a giant study with many people involved. Lev is the one who drove everything from our end, together with Andrea Di Francesco from Calico and Gary Churchill from JAX. And Lenka Donalova, you remember I showed her in the context of the exercise project. Niklas Blank is leading all our efforts in the context of stress, together with a lot of help from all the fantastic people in the lab. These are the funders, and thanks again for inviting me to be here today, and I'll be happy to take any questions. Thomas.
We do have plenty of time for a few questions. We don't have a microphone at that time, so please speak up. That would be great. So if you could just repeat the question further than
Sounds good.
That's a fantastic talk. Thank you. I have two questions. So the first one is, so as they age, the immunities go up. And I assume that the diverse genetic mouse colonies go house together. So do you see the uniqueness kind of normalized by the aging, and how does that affect the phenotype?
Yeah, really important point. We were actually really worried that an increase in uniqueness is basically associated with more mice living by themselves because their cage mates are dying. And of course, there's the known cage effect in animals from coprophagic behavior. So we were really worried about this. But then Lev did an experiment where he normalized for all of this by downsampling to the same number of mice across the entire experiment. And also by downsampling across all the different other variables like dietary restriction, and uniqueness still went up. So we don't think that it's an effect of mice living more isolated lives later in life, because if we artificially look only at mice that also live isolated lives early in life, we still have this effect. So it's most likely not driven by cages.
And the second question is that you said there's no correlation with the microbiome to longevity, but can it be an interaction between the microbiome and also the host genetics? So together, some kind of winning combination that allows them to have this longevity.
We controlled for all of this because we didn't believe the result either. We thought that there must be some kind of association, and then we controlled for all kinds of confounding effects of diet, body weight, and so on that might have negated the microbiome effect. And we looked for interactions and still no signal. So it looks like the microbiome doesn't affect how long the host lives. Genetics, of course, has a very strong effect. Genetics is actually, I didn't mention this on the slide, but if we just unbiasedly rank all the factors that contribute to longevity overall, genetics is on top of the list. There's a very strong host genome effect on how long the mouse will live.
One thing which also, this result, which obviously puzzled you also, but the mouse lifespan is very short in comparison to our lifespan. So I'm wondering whether there's something going on which is different in such a short span of time in comparison to, or do you think that uniqueness, because I would suggest that uniqueness would be incredibly high for old people. I just looked at in Switzerland, we had the last lady of 111 years old just died, the oldest lady. So, I mean, how unique would be she would have one bacteria? I don't know. What do you think about that?
Yeah, this is a great point. I just want to clarify one thing. Uniqueness is not associated with alpha diversity. So that diversity, sort of the taxonomic diversity has no bearing on uniqueness. It's just, uniqueness is basically a relative metric, right? It's only saying how far away are you from others in the same cohort. But the other point is very important. I think the differences in lifespan between mice and humans may have impacts on many of these outcomes. And we're thinking about this a lot in the context of glucose, because glucose levels, at least from this study, seem to have no impact on overall survival. But usually the detrimental effect of glucose is seen after a very long time of elevated HbA1c and glycation end products that are deposited all over the body after long periods of hyperglycemia. So it's possible that the mouse lifespan is just too short to see some of these effects, and possibly effects on cancer cancer and other things that just take a little bit longer to develop. And so I would take it with a grain of salt in terms of how we can extrapolate some of these findings to humans, because I'm pretty sure that long-term hyperglycemia has a lifespan effect in humans, which has actually been documented in many different epidemiological studies. So I think this is something we need to take into account when we talk about lifespan in mice.
Yeah, the other question I was wondering, what do you think about these nicks? I mean, you crisscross all these different mice, but how representative would they be for what you call nowadays dirty mice?
Oh yeah, great question. So this is a question we were able to answer early on in terms of what is the diversity of the microbiome in the context of a diverse host, genetically diverse host, as compared to an inbred strain. And we did some of these comparisons in terms of just looking at alpha diversity between black 6 mice, genetically diverse mice and humans, and we do see this graded effect. There's, I think, a study just about to come out from Andrew McPherson's lab, where they looked specifically at the diversity of human microbiomes, diverse genetic backgrounds in mice, and these wild mice or wildling mice that they collected from the forest or from pet stores and so on. And we don't reach the same microbiome diversity with host genetic diversity in a normal housing system. So it's basically one step along the way to human diversity, but these wild mice are a further step along the way. And of course, these wild mice are also genetically diverse. So I think we can do some interesting calculations between the environmental contributions and the host genetic contributions to this microbiome diversity. It will be extremely interesting once people start doing lifespan studies of these outbred mice with a wild microbiome. But I think the field is still too young to have any idea about longevity of these mice. This gives me a chance to present a plug for a symposium we're organizing at Penn that is inviting people from all over the country that work with these wild microbiomes. It's happening on March 6th, I think. 16th. I think March 6th. But maybe Jason can send an email invitation to everybody, because I think it's a big direction for the microbiome field in general. It's a very big direction for us at Penn, and we're trying to organize this symposium just to get a broad overview of everybody in the country who works with these mice in the forest and in these outside spaces, and basically mice with very diverse microbiomes. Yeah.
The mice that you were using, are they?
March 6th. Thanks.
Are they inbred or are they mixing in one sample?
So these are derived from 8 inbred strains, but then crossed to each other. So we generate genetic heterogeneity without losing track of where the genomes are coming from. That's why we can genotype them so easily.
Yeah, this is really great work. So one of the things that you just mentioned was you're not looking at alpha diversity, but you're still looking at beta diversity of the species that are there, right? This is done via 16S, right?
This is all based on shotgun metagenomics, but looking at beta diversity.
I think it's well known that like the types of species that are there change out, but the functional redundancy is consistent across different microbiomes. So are you looking at also the distances between the function, like the metabolic pathways, and to see if these swapped out microbes might be fulfilling a niche or fulfilling a pathway that other ones are not?
Yeah, great question. So we were pretty surprised by this the bar graph on the right here, which is basically showing the very strong age effect on the pathway level as well. So we do get very strong changes on the pathway level with age, and we actually do see an increase in functional uniqueness as well with age. So these changes that happen over the lifespan, they don't preserve the functional capacity, but the functional capacity actually evolves over the lifespan of the mice. But again, and this is something I'll show you here on the next slide. On the pathway level, there is again no agreement between different cohorts in mice and cohorts in mouse cohorts to human cohorts. So we cannot claim that there is a conserved functional signature of aging. It basically behaves just like the taxonomy, where we get this random evolution over the lifespan with no apparent determinism in terms of the age of the host. Oh, in the chat. Maybe you have one more question then.
You may have meant this, but if you were to probably sex biological factors, no differences between males and
males or?
Yeah, this is a really critical question. The entire study was done on females. And the reason why we did it on females is because when you calorically restrict male mice, they start fighting over the limited resources and they lead to unnatural death, basically. So this study on caloric restriction over the lifespan can only be done on females unless you single house the mice. But then single housing 1,000 mice over the lifespan is crazy. So that's it's a big caveat because all of these conclusions here I should have said in females, because we don't know if male, we definitely know that there are sex differences in longevity and dietary interventions. Whether they affect some of these pathways I just talked about, we don't know because it was only done in females. Oh, I just see that my laptop has 3% battery left, so we very quickly answer the last question. Which is, I don't know if you can see it on the screen, but the question is from Masaki, who asks, regarding the diet experiment, what about the starting age of the experimental group? Does it change if you start dieting at an older age or if you make it a habit from birth? Yeah, this is a really important question. I should have said that we started the dietary intervention at 6 months of age because we wanted to avoid basically dietary effects on adolescence and mouse growth and so on. You may remember this this curve I showed with the different ways. So the dotted line is where we started the intervention. You can see that, of course, the growth trajectories are all overlapping until then, and then they diverge. But it's a very good question in the context of this study I introduced here, which is basically saying that you get lifespan effects even if you start dieting very late in life, or if you're dieting for the majority of the life, but then you switch to ad libitum. So this is a very important question that we didn't cover because we all, we started the dietary interventions all at the same time at 6 months of age, trying to avoid these basically juvenile effects of caloric restriction. But it's completely possible that some of the conclusions would have changed if we had started, dieting the mice when they reach peak body weight, for example. But we had to take one guess in a big study like this, and we decided to do 6 months. Great. One more question.
Yeah, so just looking at all these results, how did you change your lifestyle?
You can purchase my dietary plan outside in the lobby after the lecture. But to be completely honest, after seeing the first results of this study, I started doing this 16-8 method, which is here in the top corner, which is very compatible with normal life. You basically just need to skip breakfast. But at least the animal data is extremely convincing. We've done a few experiments with intermittent fasting in the lab, which I haven't shown you here. And also the human study situation is pretty convincing. So it's definitely something I can recommend for everybody that is interested in trying something.
All right, well, before his computer dies, let's thank Christophe.